Okay, good afternoon, everybody. I'm going to talk about Contactin-1 and other paranodal proteins in inflammatory neuropathies and in particular in CIDP. What is CIDP? CIDP is the acronym of chronic inflammatory demyelinating polyradiculoneuropathy. Is the most frequent chronic inflammatory neuropathy, but it still is very rare. It's a disease that is chronic or multiphasic. Symptoms progress along 8 weeks or more. This is very important. The diagnosis is based on clinical and electrophysiological criteria, and there are no biomarkers so far that could help in diagnosis. It's pretty disabling, despite there's treatment for this disease. 50% of patients remain with significant disability even with treatment, and response to immunomodulatory therapy, and in particular to plasma exchange and intravenous immunoglobulins and steroids. And it's very heterogeneous disease. There are several clinical variants. The typical CIDP is a sensory motor, a predominantly proximal disease with demyelinating features in the electrophysiological studies, and with high protein content in the CSF. But there are a number of other clinical variants ranging from focal presentations, pure motor, pure sensory, or pure ataxic presentations, distal variants, different associated features like cranial involvement or monoclonal gammopathy, and various presentations. It can be relapsing remitting, it can be chronic progressive, so it's pretty heterogeneous in its presentation. It is presumed an autoimmune origin, and this is the type of cartoon that everybody presents to show the pathogenesis of this disease, in which it is supposed to have the sensitization of against an antigen in the periphery that promotes a T and B cell response that cross-reacts with a myelin antigen in the nerve, and that drives the disease. But this is based almost exclusively in animal models, and there is there's not much research with samples or with patients. It is autoimmune because, or at least it's presumed autoimmune because we know that it responds to immune modulation or immune suppression, because we can find inflammatory infiltrates in the nerves and roots, and necropsies and biopsies, and also because some experimental evidence shows that there is an autoimmune process in its pathogenesis. And it has always been believed that there is a prominent role in its pathogenesis of autoantibodies. First, because this disease responds pretty well to IVIG and plasma exchange therapies. Second, because it has been found the position of IgG or IgM in sural nerve biopsies, because this disease associates with polymorphisms in Fc gamma receptors of immunoglobulins, in particular with this one, with FcγR2b, and because, and most importantly, because passive transfer of serum from patients leads to a neuropathy, inflammatory neuropathy, in animal models. The topic of autoantibodies in CIDP has been a hot topic, and probably the only hot topic in this disease for more than 2 decades, almost 3. But the description of antigens or potential candidate antigens followed the development of animal models. Considering the demyelinating nature of the disease, most studied antigens were myelin antigens, in particular myelin protein 0, 2, 22, and gangliosides. And none of these antigens, the antigens described so far, had any clinical utility because they were present in a significant proportion of controls too, because they were discovered with techniques that didn't take in account conformational epitopes, and also because the approaches, the experimental approaches, were biased, were mostly candidate antigen selection of the antigen studied. Since a few years ago, the notes of Rambier that had been studied before, but they are now again coming up all the time. Notes of Rambier have been found to be disrupted in the biopsies, nerve biopsies of these patients with inflammatory neuropathies, in particular in CIDP, and their structure disorganized. In particular, the group of Claudia Sommer very recently has shown that it looks like the demyelination process in these patients, or at least in a subset of these patients, starts from the paranodal areas, and depending on the disease is different, but in CIDP it looks like it's pretty specific. It starts with an elongation of the paranodal gap. And in this paranodal area, there are a lot of proteins, but not only, and this paranodal area is pretty important for axoglial junctions. So a demyelination process not necessarily has to come from an antigen that is in the myelin, in the Schwann cell, but also could come from an antigen of the axon. So that led us to think that maybe the right approach was not taken so far, and to think if maybe axonal antigens could be involved in the pathogenesis of this disease. And this led to this paper that is the one that was awarded yesterday. And in this paper, what we did was to take 46 patients from our clinics with CIDP, typical or atypical CIDP, that were followed in our clinics, and so we had clinical information available, and followed an unbiased approach for antigen discovery. What we did basically was to take, in this case, primary cultures of rat hippocampal neurons, and we chose hippocampal neurons because first they had been used before for antigen discovery in other diseases, in autoimmune encephalitis, and they are pretty clean and pretty good for immunoprecipitation studies. Second, because the machinery, the molecular machinery of myelination is pretty similar both in the central and the peripheral nervous system, and we had them running in the lab, so we used this model. If patients reacted against neurons by immunocytochemistry, what we did next was to immunoprecipitate with patient sera with hippocampal neurons, and then the precipitate we ran in an electrophoresis gel, and if there were any differential bands between patients and controls, we excised the band and sent to mass spectrometry. If any protein was considered relevant, then what we did was to confirm the reactivity in a different model in immunocytochemistry with hex cells transfected with a protein of interest, and then confirm if there was any reactivity against relevant structures, in this case in peripheral nerve, in rat teased nerve fibers. What we found was that 7 out of 46 patients reacted against hippocampal neurons. That was just 15% of the total cohort. And 4 patients reacted very strongly, and those were the ones we used for immunoprecipitation experiments. So of those 4 patients, 3 of them had a very clear differential band in the electrophoresis that was excised and sent to mass spec. And those 3 patients identified Contactin-1 in the immunoprecipitation, and one of those 3 patients also precipitated this other protein, Casper-1, and this was confirmed in this 3rd patient by Western blot, that in the precipitate both proteins were present. So then we moved into immunocytochemistry to confirm the results. We found that of those 3 patients, 2 had antibodies against Contactin-1 transfected HEC, and the other patient did not react when only Contactin was transfected, but reacted when both Contactin-1 and Casper-1 were co-transfected, and only in that situation. And that this was the patient that precipitated both Contactin-1 and Casper-1. So from the controls, we had 104 negative controls that were of different diseases, all of them or many of them of autoimmune origin, and also resemblant to CIDP, or at least with some features that could be confused with CIDP. And all patients and controls were negative for Casper-1 antibodies when Casper was tested alone and not in combination with Contactin-1. Also, to further confirm the specificity of the reactivity, what we did next was to pre-incubate the serum of the patients with either HEC cells, untransfected HEC cells, or Contactin-1 transfected HEC cells, and we saw that the reactivity was abrogated when the serum was pre-incubated with Contactin-1 transfected HEC cells. And well, here is the combination of images. And the most important part is that these three patients shared the clinical phenotype, the clinical features. They all were older. The mean age was 51 for anti-Contactin-1 negative patients and 71 for these three. They had a motor predominance instead of sensory motor. They had an aggressive neuropathy, and in fact, two of them had been diagnosed as EBS, which is a very close related neuropathy. Two of them, well, all of them were bed-bound or wheelchair-bound very, very rapidly. And this is something that is rather atypical for CIDP. They all had axonal damage at onset. They had typical demyelinating features as other CIDP patients, but they also had acute denervation in the first EMG that was performed. And finally, although most patients or the majority of patients with CIDP respond to IVIG, these patients did not respond or respond very poorly to IVIG. So what's the relevance of these findings? The antibodies against the Contactin complex identify a homogeneous CIDP subgroup, and then these autoantibodies are the first biomarker in CIDP that can have diagnostic and prognostic implications. And finally, that the screening or the screening technique with neuron immunocytochemistry and identification of antigens by immunoprecipitation proves as a useful approach for antigen discovery in inflammatory neuropathies. So we know that those antigens or those autoantibodies are specific, and we know that they can be useful in the clinics, but we don't know if they are pathogenic. These studies are following, but only seeing what coming back to this slide, this is where Contactin and Casper are, and this is where the demyelination starts. So we believe that although we have to confirm this, these autoantibodies are pathogenic. And also, if we look at the Contactin-1 knockout mouse, we see that it has slow conduction velocities. That is something that our patients have. Prolonged latencies, that is something that our patients have, and decreased amplitudes that also our patients have. But these are the patients that were very positive for neurons, for hippocampal neurons, but almost 41% of patients react against other nerve structures, nodal or myelin structures, and also some patients that didn't react against hippocampal neurons had a paranodal pattern of staining. So, and we also knew, because simultaneously to our study, other studies were published in other proteins, that one of the antigens that had been described at the same time as ours was neurofastin in this type of neuropathies. And neurofastin has two isoforms, one that is in the node and clusters sodium channels in the node, but also neurofastin has another isoform that is in the Schwann cell and the and that binds to Contactin-1 and Casper. And what we did was to just check if any of our patients had autoantibodies against neurofastin, and we did immunocytochemistry with both neurofastin 155 and 186 with the immunocytochemistry technique, and we found 2 patients that were positive for anti-neurofastin 155 antibodies, none for the other isoform, and also that these patients were in fact the ones that reacted against the paranode but not against hippocampal neurons. So these are the clinical features. I don't know if I can you switch on the video, please? So this is just to show you how it looks like in the clinics. Well, we are going to wait a little bit, but you can see that, well, the patient first has distal weakness and a tremor that you will see better now. This is not the typical CIDP at all. Now you will see. Inflammatory neuropathies can have tremor, but usually not that amplitude and not that frequency. I will explain you later what we think. So this is to show that the patient had a prominent distal weakness. He cannot raise or lift his foot up, while the typical CIDP also is usually proximal, not distal. And the tremor, you can see that the tremor was also present in the legs, although it was less prominent or had less amplitude. And let's see. And this is how the patient walked, just to show you that the distal weakness, you can see that the patient doesn't lift his foot at all. And this is an inflammatory neuropathy, but it's not the most typical CIDP. So the weakness was predominantly distal. The patient had ataxia, a pretty disabling tremor of low frequency when tested with electrophysiology was 3 hours. In this case, the EMG was demyelinating. It had not axonal features. And again, the patients did not respond against IVIG or steroids, but responded to plasma exchange. These were 2 patients, remember. So what we did next was to ask other people in the CIDP, in the CIBERNED network, using the databases that Isabel has set up. And we asked for other patients that were IVIG resistant, considering that it's not so frequent that patients are resistant to IVIG. We asked for IVIG resistant patients, and we found 8 patients coming from Valencia, from Belviche, from Santiago, from Marqués de Valdecilla and Santander. And we found 2 more patients with auto-antibodies against neurofascin 155. And this is one of the patients coming from Santiago de Compostela. You can see that it's pretty similar to the other one. And there was another patient from Santander that had similar clinical features despite the absence of tremor in this case, but also no response to IVIG, distal involvement, etc. So this is the patients. And then when we had the 4, we confirmed the reactivity with ELISA, and we showed that, as expected, the patients with antibodies tested by immunocytochemistry had higher titers. In fact, all the other patients, CIDP or controls, did not have any antibodies against neurofascin. And very important in this case, all patients had antibodies or predominantly antibodies of the IgG4 isotype. And this is important because IgG4 is a special immunoglobulin. IgG4 does not bind complement and does not bind to Fc receptors, Fc gamma receptors, which are 2 of the targets of the IVIG. And that could explain, at least we think that that could explain why these patients do not respond to IVIG and do respond to plasma exchange. So to go further, as I said, tremor is a common feature in inflammatory neuropathies. It can almost affect 50% of patients, but this type of tremor is not so frequent, and in fact in our series there was no other patient with that tremor. Very recently this group published a paper saying that the cerebellar learning distinguishes patients with CIDP and other inflammatory neuropathies that have or not tremor. So suggesting that the tremor in these patients, despite historically was thought to be of peripheral origin, was in fact of cerebellar origin. That is something that led us to think a little bit more, and in fact this group say this phrase, that is that one possibility in these patients that have tremor and cerebellar impairment, one possibility is that those with tremor have a specific antibody involved in causing the neuropathy or cross the blood-brain barrier. And they go on like this. And so we decided to test if that was true. And in fact, as expected to, these patients had not only features that suggested a cerebellar origin, but their antibodies bound to, this is the patient, this is a control, bound to the molecular and the granular layers of the cerebellum, while the controls did not. They also bound to other structures in the brain. And this was true for all 4 patients, even for that patient that did not have tremor from Santander. And the problem with that patient is that the titers of the antibodies were lower. So we believe that this hypothesis that the other group prompt of maybe the antibodies crossing the blood-brain barrier, we believe that could be the case that explained why just that patient did not have tremor and all the others had a tremor of cerebellar features. And so antibodies, to sum up, antibodies, IgG4 anti-neurophasin antibodies associated with a homogeneous CIDP phenotype, again, with distal predominance, tremor, IVIG resistance. The tremor in these patients, we believe, has a cerebellar origin because the tremor features itself, because the antibodies bind to cerebellar structures, and because the model of the animal model of knockout of neurophasin 155 shows not only a demyelinated neuropathy, but also cerebellar impairment. And although the overall prevalence of these antibodies is low, the same as Contactin-1, it's significantly higher, up to 25% of those patients that do not respond to IVIG. So in conclusion, autoantibodies against node of Ranvier structures play a role in CIDP development. The CIDP clinical heterogeneity that I showed in the first slides disappears when patients are grouped, defined by autoantibody status and not by clinical and electrophysiological features. Anti-Contactin-1 and neurophasin 155 antibodies are the first autoantibodies with diagnostic and prognostic implications in CIDP. And these findings, we believe, will help personalizing CIDP therapy according to the presence or not of these autoantibodies. And just a little bit for the future, not only patients have antibodies against hippocampal neurons, but we are starting to see that they also can have antibodies against Schwann cells and DRG neurons, which obviously will keep us busy. I would like to thank, first of all, to Isabel, because this is thanks to her, and also Gisela Nogales, that was a close partner and co-author of my paper. So thank you.
Questions?
Yes, I can.
Did you check, I don't know if I missed idea of the antibodies of the patient, the first ones for Contactin-1, Casper-1, that could not be affect, or at least under high titers affect Contactin-2, Casper-2, that are at juxtaparanodal. Because that would be a nice picture to show that an initial delamination or separation of the paranode could progress to the next, and so explain a little bit.
We did it, we checked if these patients had, because this was a question for our reviewers in the paper, and we checked if the patients first had if they had autoantibodies against Casper-2, because that was described in neuromyotonia, and it's not the same disease, but it's also peripheral nerve, and they don't have autoantibodies against Casper-2. The second thing we did was the opposite, to see if a patient with anti-Casper-2 antibodies and neuromyotonia, the only patient that we have because it's extremely rare, if that patient had antibodies against Contactin-1 or Casper-1, and that patient did not have antibodies against Contactin-1 or Casper-1. So it looks like it's very specific.
And just the second, for a curiosity, for these patients with so strong tremor clinically, did you check for potential abnormalities in the muscle spindles and gamma motor neurons? That could be explained a lack of control, peripheral control, not just going to the cerebellum.
Yeah, these patients for sure will have a peripheral deficit. In fact, they have sensory deficits, and they have an ataxia that also worsens with a little bit with eyes closed. But this is something we see in many patients with CIDP, and in fact we have pure ataxic CIDPs that have a tremor also, but the tremor is of a higher frequency and not that amplitude. So of course they could have also a peripheral component of the tremor, but we believe that this has a prominent cerebellar features, and that could be the explanation why. There's another question there.
Rafa tiene otra cuestión.
No? Okay. Very beautiful. So one thing, I'm not sure that I got it right. So it's regarding the phenotype or the symptoms of the patient, the one with the contacting antibodies. Do they have central symptoms or not?
They don't. The Contactin-1.
Please comment on that.
Yeah. Well, this is something that happens all along neuroimmunology. For example, aquaporin-4 neurooptic neuromyelitis.
Yes.
Aquaporin-4 is all over, and mostly in kidney, etc., and they don't have kidney problems. So we don't know exactly what's going on, and we don't know if the blood-brain barrier is not so affected to have central symptoms in these patients, or if there's anything else that you need, for example, complement that you don't have in the central nervous system for this specific type of patients, not for the neurofasting. But the thing is that they don't have symptoms. And that also, as I say, happens in other diseases. We don't know why, but it's the case.
Okay, good.
Yeah, very nice, your paper. But let me ask you two things, because I was surprised you call that the antibodies are pathogenic. There is any evidence for that? For example, do you see change in the antibody titers with, say, progression of the disease, or there are stable titers that it behaves?
Well,
always there?
We believe that they are pathogenic. We don't know. We have some experimental evidence that the antibodies disrupt the binding of Contactin-2 neurofasting. So, but this is not published, and this is very preliminary, and we didn't want to show it specifically. We don't. So these patients are very few, so we cannot make correlations about the clinics. We could, at least for the neurofasting, those ones, it seems that the more severe the disease, the higher the titer. But you cannot do statistics on that because it's just 3 patients. What we know also is that when you treat, for example, the first patient that we got of neurofasting 155, the titers decrease when you treat the patient and the patient improves, but you cannot say if that's a direct cause relationship, but just a epiphenomenon. The general improvement just drives the antibodies down.
Another question, have you checked if there is a T-cell response?
Sorry?
A T-cell response, a proliferation of CD4 or CD8 in response to your antigen, the antigen you have identified in those patients?
No, we haven't.
Okay, thank you.
To get at the pathogenesis, are you aware if anyone's actually tried to immunize mice with antigens to these to see if they develop a neuropathy?
Well, we have to, this is, in fact, this second thing is not published yet, it's under review, so I'm saying a lot of things that are not published, but so yes, we are collaborating with a group in Marseille that are basic scientists that have animal models and everything. And we have 2 plans. First, active immunization, and second, passive transfer. We have plasma from the plasma exchange of these patients. We have liters of plasma of these patients, and we believe that we could sometime demonstrate that those are pathogenic.